Thanks for inviting me and thanks for organizing this webinar. Um, so today I wanted to first introduce what are REEs, rare earth elements, and then look a bit at how to work with northern communities, because many of the mines that will be exploited in the future are located up north. And after that, I will give you some results that we've gathered over the last 10 years on monitoring REEs in northern freshwaters, using lichens as atmospheric REE biomonitors, also using zooplankton as aquatic biomonitors, and also we are now developing amphipods for sediments, but I won't present these data today. And we'll look also at how REEs may be bioaccumulated in animals and within cells, also how they partition within cells. This informs us on toxicological impacts, potential toxicological impacts, and also on trophic transfer of REEs. Then we'll move into food web transfer of REEs, and a bit of an extra topic at the end, what happens to REEs when you cook country food, because I wanted to also to really touch the issue of country food in general. So rare earth elements are essentially a series of 15 metals, the lanthanides that are found at the bottom of the periodic table, plus yttrium and sometimes scandium as well. And so you can see that there, in this lanthanide series, there is a gradient of abundance with the light rare earth elements being more abundant than the heavy one. Also, most of these REEs are trivalent, so that's quite important when understanding bioavailability, for instance. And there is a gradient in atomic radius that modify a bit the properties of these elements towards, for instance, particles. So usually the light REEs are more particle reactive and therefore may be more easily scavenged from the water column towards the sediments than heavy REEs, and this is partly due to the change in the atomic radius with atomic mass. In general, there is a big covariation of all these elements together in the environment. And to prove that a bit, I've taken a figure from one of our first papers on REEs that we've prepared about 9 years ago, and you can see a very strong relationship between the different matrices. So these are samples taken from different lakes in Quebec, Canada, from the water, benthic invertebrates, fish, sediments, zooplankton, and you can easily see, for instance, that cerium is really well correlated with lanthanum with a correlation of 0.99. So really strong correlation across matrices, across these elements. So that's a first message for this webinar, is that they are a family of contaminants or potential contaminants that are being co-transported in the environment, and when you are exposed to one, you are exposed to many. So many bioassays are made with only one element, but in reality you should consider the fact that often they move together. But since there is a difference in abundance, usually if you look at the first ones on the left side here, you will get most of them. But it's important to consider mixtures in this instance. They have many applications, as you all know. They are in our computer screens. They are in the medical sector. They are used as contrasting agent. For instance, gadolinium is often seen in the wastewaters of cities because of hospitals. They are also very important in the energy transition. They have very important properties as permanent magnets, and so they are used in wind turbines. They are used to build electric cars as well. So the global demand is really increasing these days, and the market has been mostly dominated by China, but because of geopolitical tension and also some restriction to exports by China, there is a pressure to develop the mining industry in many countries right now. And that's the case for Canada. We'll mostly talk about Canada today because that's where I've collected my samples most of the time. And there has been a first mine open in 2021 in the Northwest Territories, in the region of Yellowknife that John knows very well. It has opened in 2021, but it has had some issues. It closed at one point. But in general, in Canada, there is not a lot of mining going on at this point in time. There are many projects, and there is a lack of environmental data in general for these elements. And so we are in the process of developing guidelines at this point. So on the left side here, you can see a pie chart of the global RE projects, and you can see that there is a diversification of those projects, and China still dominates, but Canada has many projects. These are not mines that are actually active at this point. These are projects. And on the right side, you can see RE deposits across Canada, and here the message is mostly that it's really not concentrated in the south. There are many instances where the deposits are more northward, and so they will be placed on indigenous land of the Inuit peoples often, or of First Nations as well. And that's a part of the seminar I wanted to also emphasize is the relationship with northern communities. And I will describe a case study where it started with people from a university in southern Quebec called UQTR. So it's University of Quebec in Trois-Rivières. And they kind of had a very different approach to what people are usually doing as scientists. So they sent letters to 14 Inuit communities asking them what are their concerns regarding environmental changes and how they can help. So it was really turning the table a bit. And one of the community, led at the time by Hilda Snowball, who was the mayor, but now is the chairperson of the Kativik Regional Government, so the entity that governs Nunavik. This answered the letter and said, we have a project for a rare earth element mine in our river basin, and we are concerned about that. So we would like to have more information about that. And that's the point where I've been contacted by the team because I had an emerging expertise in rare earth elements at the time. So this project is called Imalerigit, which means people who study water. And it's a community-based monitoring program that has been going on since 2016. It's led by the community of Kanjiswalujjuaq and UQTR, and other universities have joined, such as my university, University of Montreal. One very strong part of this initiative is a one-week expedition every year on the land where you have elders sharing knowledge with youth, but also scientists. So you have a combination of expertise from indigenous knowledge and science knowledge being shared together about the land and including rare earth elements. And also the community indicated that they wanted to have a data set from that they could own and that they could use to monitor what will happen with the exploitation of rare earth elements in the watershed of the big river there. It's called George River. And this initiative was really successful and got the Arctic Inspiration Prize in 2018. So it's a community-based environmental monitoring program in Nunavik where many samples are taken from the land. You have lichens to monitor atmospheric deposition, sediments, water, biofilms, and also animals collected by people from the community. Many of these animals are consumed by the community. It's still a community that relies on country food quite a lot. It's still there's a problem of shifting diet towards Western diet, which entails issues of calcium deficiency, iron deficiency, diabetes. Many health issues are related to the fact that they are kind of not using their country food as much as they did in the past. So They collect animals for us, with us, and they fill up these forms where many details are indicated so that we can then use these data for scientific purposes. And the land camp has been very successful over the years. So here you have the photos of the groups since 2016. The only date that is missing is 2020, as everybody knows why. And now we are the team is co-constructing funding applications. So in the start, it was mostly led by scientists, but the last few funding applications are led by the community. And you have here an example of one of these workshops where ideas of about environmental change in the area are discussed and they are prioritized. And the end product is this. This is part of the applications that have been successfully submitted recently. And you can see on the external ring here the most critical concerns of the community. And then the pie chart is divided into what's feasible and what's not feasible. And for instance, you can still see that among all the environmental issues that they have with climate change, for instance, they still indicate that critically they want to know more about air quality and dust particles from the mine, the future mine, and water quality upriver. That's also where the mine is. So, and they are still very concerned about this after 10 years, and we've collected data over this period. And now there is also capacity building within the area with the Nunavik Research Center. They will acquire an ICP-MS there, and they will be able to monitor by themselves rare elements. So they are currently in our labs to have some additional training on how to process samples so that they can really implement their method in up north. So now we'll turn our attentions to some of the data we collected. Many of these data were collected as part of partnerships with Environment Canada and also with different universities. So in general, the framework is to work on all the data for critical metals. So it's not just rare earth elements, it's also platinum group elements and many others, lithium and so on. And we look at the speciation of these elements very thoroughly, association with organic matter, inorganic ligands, and then we look at how they can be bioavailable towards our organism using a conceptual framework such as the free ion model or the biotic ligand model. Then we look at what's happening internally within the organisms up to the cells where we can do some subcellular fractionation and look into the accumulation of these new these data pore elements into different cellular fraction in order to, at the end, derive guidelines. So first of all, I wanted to talk about our multi-year monitoring of RE elements in the water, and I will only touch the freshwater today. So there are many studies right now from marine systems, but I won't discuss them. So we've been collected samples from freshwaters over the last 10 years in many regions, like mostly in along the St. Lawrence River and its tributaries, the Bay James rivers, the Northwest Territories that are a bit out of the map on the west side, and then the system that I want to discuss right now is the George River and the Korak River system. It's like up north there, near the city of Kangiswalujuak, that is about here on the Angava Bay in northern Canada. It's in the subarctic. So because it's in the subarctic, it's in an area of great transition. So you have in a couple of 100 kilometers a shift between continuous, extensive, discontinuous permafrost types up to really no permafrost in some instances. So it's really a great environmental gradient here happening. You have the mine that is located here, the river that goes from south to north and goes into the Angava Bay. And we also looked into a second watershed, the Cork River watershed, that is a bit different with respect to permafrost. It's richer in continuous permafrost there. And the data that we collected over the last 10 years there, and partnership also with the Ketivik regional government and Nunavik parks, are shown here. And a point that I wanted to make here is that REEs often travel together, but also they are found many times in deposits that are also rich in thorium and uranium. So a big problematic link to REEs when you are mining it is the release of radionuclides. So that's often something that is of greater concern than the REEs themselves. So it's really important to monitor thorium and uranium at the same time. And here you have a series of aquatic systems in the Nunavik region of Kangiswa-Lutjuak, including the place where the mine is located. So you see here Lake Brisson is the lake that is just beside the exploration camp for the mine. And what's striking is that the levels are really low, including near the mine at this point in time. So it will be kind of easy to see any disturbance of what's happening there after mining because the levels are initially low. And this is another important point to make is that it's not because you're near a mining site or a geologically rich region with a lot of REEs that the water will be rich in REEs. REEs are often precipitated in water if there's a bit of phosphate or if the pH is too high. So there are many instances where you may have low levels in rich regions. And that's what we've seen near the Nichelacho mine in Western Canada. The levels in the water are extremely low. So for instance, here, Lac Brisson is near the mine, but it has a pH of 7.4 and a concentration of organic carbon of 1.9. So a bit of an alkaline pH and low DOC, that makes it low in REEs. Whereas you have thermokarst lakes that are really pristine, but they have an acidic pH and higher DOC, and they can therefore keep more REEs in the water column, and the levels are higher there than in the near the mining site. And usually you can see that thorium and uranium roughly follow the REE levels as well. We often present the data of REEs as combined or summed. That's what I've done most of the time in this presentation, but we could do lanthanum, for instance, or cerium. It would give very similar trends. We compare these concentration in the water to others found elsewhere in the world. So here you have levels from northern Canada, southern Canada, France, Brazil, and some mining sites in China. This is a log scale, and you can see that the sites in China that are actively mining are quite high compared to the others. And with respect to the subarctic here in northern Canada, you can see that the levels are still like they're ranging over a couple of orders of magnitude. But the sites near the only active mine in Canada are the lowest, like a very, very low because of water chemistry. We tried to find out more about the drivers of these concentrations, and the best relationship we got from the north were related to dissolved organic carbon, as I mentioned before. So about 40% of the variance could be explained by that in our systems. And then aluminum and iron are often correlated well. In certain places, it's really strong, the relationship. Here, it's a bit weaker, but still there for the dissolve phase. So usually we think that there's an association with aluminosilicates in the water, with iron oxyhydroxides in the colloidal phase mostly. And so there is a combination between these elements for co-transport, we think. And as we've seen in the last graph, there's also a correlation with the radionuclides. So you have in gray the uranium trend and in red the thorium trend there. Quite unexpectedly, we also found a relationship with temperature. So over the course of many years, we encountered many different environmental conditions. So overall, we had a range of temperature from 3 degrees Celsius to 20 something. And across this gradient, we could see a decrease of REEs as a function of temperature. We are not totally sure what it means at this point. We think it's related to the PCO2 of the water, the effect of temperature on the carbonate equilibrium, because rare earth elements are often bound to carbonates, so that could explain it a bit. But we will look into that in the future, but it was a nice trend to see in the context of climate change. Now I've kind of modified my presentation to put more emphasis on biomonitoring because it's AMAP. Also, it's organized partly as a part of AMAP. So I've chosen to talk about lichens a bit more because we are looking into them as possible biomonitors of atmospheric deposition. And we've collected these lichens across the George River system and the Correc system with the help also of the Nunavik Research Center, who were able to collect many samples near the Strange Lake deposit, so near Lake Brisson here, where the mine will or may eventually be built. So we divided them into different taxa, and we tried first of all to test if there were good biomonitors. And the way we did that was to see if they were reflective of the concentration in the soil. Like if they are directly related to concentration in the soil, they are not so good monitors of the atmosphere. And what we found first is that there was not a big relationship between REEs in the underlying soil where they were collected and REEs in lichen. What we wanted to see is a relationship with other tracers of atmospheric deposition, and we chose aluminum as an example. So there was a very strong relationship between aluminum in the lichens and REEs in the lichens. So we think that lichens may be a good biomonitor of REEs in this environment, and that's what we will suggest in the future. These are unpublished data at this point. We plan to publish them in a couple of months. For the aquatic side of things, we tested if zooplankton could be a good biomonitor of REEs, and we are now developing amphipods for the sediments, but I'm not presenting these data today. So for the zooplankton, these are data from Gwyneth Macmillan, and she collected samples from 39 lakes across 5 geological provinces, from Montreal here to Kuujjuaq in the subarctic, through Iqaluit and Resolute Bay in the high Arctic. And here, just a word about the conceptual framework where we're working. Like usually you have a metal in solution and it will partition between different ligands. Some of it will be complex to the organic matter. Some of it will be complex to inorganic ligands. And some of it will be free as an hydrated free ion. And it is postulated that this free ion is the best predictor of what can be accumulated by an organism. So that's called the free ion model. There's also the biotic ligand model that considers competition at the uptake site by some ions in solution, such as calcium and protons. I won't talk about the biotic ligand model further, but we'll look into the free ion model. So we first correlated surface water of lanthanum concentrations in surface water to zooplankton concentration, and found a relatively satisfying regression with about 43-44% of the variance explained. But then we calculated the free ion. Like you cannot really measure it, it's really low. And so what we do usually is to use a thermodynamic model. It's called WAM. It's the Windermere humic acid model. And it calculates, based on a series of affinity constant, the concentration of free ion in solution. So we did that, And when we plotted that as the x-axis, we strengthened the correlation quite a bit. We were able to explain 72% of the variance in this case. So that tells us that the zooplankters can really be good biomonitors of the aquatic water column for rare earth elements, and we can understand kind of what are the principles of the uptake through the free ion model. Now, how do these RAE make their way up the food chain, and mostly like in food that people consume? So first of all, we wanted to know about fish, like what's happening when these fish will be exposed in the future to high levels of RAEs. And so we've done these experiments in the lab with model species used by the government. So an experiment here was to submit a trout to either water as a source of yttrium, which was our model REE, or to food. So for the food, we contaminated the Daphnia mania with yttrium, and then we prepared food pellets and fed it to the trout. And then that way we could compare the uptake from either the water or the food, and we found out that the uptake from the water was far greater than the uptake from the food. So it seems that food is not such a great flux towards the trout for uptake of yttrium. And then we looked at what was accumulated in the trout and found out that most of the yttrium was accumulated in the gastrointestinal tract, then the gills, then the liver, and at last the muscles. So it means that the muscles are really bad at taking up yttrium and probably other rare earth elements. And this is consistent with the data I will show now on real animals from the wild from Nunavik. So this experiment showed us that the water uptake is an important route, and muscles are not great bioaccumulators, and livers are better accumulators. And with the Kangiswalujua community, we collected many Inuit country food. So on the left side, you have a table where you can see these different organisms that we've selected. And on the right column, you can see whether or not the community is consuming a given organ. So for instance, for the seal, you have the muscle, the liver, the blubber, and the bone, and all are being consumed. But in contrast, the sculpin, which is a fish, is it's only the muscle that are consumed. So we measured the REEs in all of these organs, and we won't go through all these graphs. The general conclusion from these graphs is that usually the liver is the best accumulator of REEs, nearly as much as the kidneys, and then you have the bones, the muscle, and the fat. And this is important in deriving exposure and risk for the consumer. Usually the muscle is really at the end of the sequence, as we've seen in the experiment. Now we'll dig in and look at what's happening within the cell. So this is important in order to understand how organisms are handling rare earth elements. When they are exposed to them, what will they do with them? And we've developed this technique with my colleague at UQAM and others before, where you can separate a cell into its component. It's not as nice as this graph, like you are not able to separate all these subcellular fractions, so there are limits. We are using centrifugation and also some other methods to separate these fractions more or less efficiently. And the idea is that if a contaminant accumulates in an inert fraction of the cell, such as mineral concretions, it won't be a problem regarding toxicity. It will be sequestered there. It's probably a detoxification process. But if you have a lot of contaminants in mitochondria, that's more of an issue because it's so important for the cell. So we've done that on 3 organisms that are used as models, like it has been mostly done by Pierre-Yves Cardon, a PhD candidate that we had with Michael Rosabal and Claude Fortin. And so Pierre-Yves decomposed the cells of these individuals, and so he separated the sensitive fractions, which included mitochondria, organelles, and enzymes, which were separated as heat denatured proteins. And for the detoxification process, it was the metal-rich granules and the metallothionein-like proteins. And so the results in general are the following. So first of all, we can see that the big numbers here are the bioaccumulated yttrium at the end of the experiment, and you can see that very easily that Daphnia mania is a far better bioaccumulator of yttrium than the trout, for instance. You have orders of magnitude of difference. And then if you look at the subcellular fractionation of this yttrium, you can see that in Daphnia mania, it's mostly in the detoxified fraction, so granules or maybe the exoskeleton as well. Whereas in the trout, it's mostly in sensitive fractions such as mitochondria. So it means that you have very opposite ways of handling yttrium in nature. You have some organisms that are really good at accumulating it, but they are partitioning it in inert fractions. So it's maybe not such a big deal for them. And you have other animals that are able not to accumulate it so much, but what they do accumulate goes in sensitive fractions. Now, just a bit of a word on what's happening when REEs are going up the food web. So we've done this type of graph for many sites, and we've done also some graphs with stable isotopes of carbon and nitrogen up south. But in the north, these are the data that we've collected. But all the data that we have from our labs is indicative of not a biomagnification. It's never biomagnified. It's also always biodiluted. And here are examples from terrestrial, freshwater, and marine ecosystems. For instance, you have the lichen here at the base of the food web, and then you have some birds, the Canadian geese and the ptarmigan that are lower. And in this instance, we even have the crop content from the ptarmigan. So really what it is eating, and then what it is accumulating. And we can see that it's really decreasing. And for freshwater, we have biofilms, benthic invertebrates, then Arctic char and whitefish. So again, when you get to the fish, the levels are really low, and note that this is a logarithmic scale. And then in marine systems, you have algae, mussels, fish, and then ring seals. And again, it's going down. So really, I'm quite convinced that biodilution is occurring trophically for RAEs in nature. And the last part of the talk was about country food cooking. So maybe that's a bit different from what is usually presented. So country food is really important culturally and for the nutrition of these population. There is an important issue of food safety because some people do not eat as much as they want. Like in recent surveys, people from Nunavik have declared that they are missing days for food sometimes, and meals like not 3 times a day all the time. So part of the population is really having a hard time to get food, and country food would be a really good way to increase their food intake and prevent deficiencies, and it's culturally important. So it would be nice to be able to promote country food if it doesn't have too much risk associated with it. And current risk assessment do not consider changes in metal concentration or speciation during cooking. But in recent studies, we've shown that for mercury, for instance, if you cook it, it will usually increase the concentration because you lose some water and the mercury stays in the meat. So at the end, you are concentrating the mercury in the meat that you've cooked. And we wanted to see if that was the case for other elements. So that's what we did. We did an experiment where we cooked whitefish mussel and gray seal liver, and we looked at the partitioning of different metals of the periodic table between the cooked meat and the cooking juice. And the hypothesis was that the mercury and other class B metals that have a tendency to complex with proteins would stay in the meat, whereas others like alkali metals and would probably get out, as previous studies have shown. And since rare earth elements are considered class A metals, so mostly associated with oxygen complexes and not so much with nitrogen or sulfur complexes, we anticipated that they will move out of the food during cooking. But contrarily to our expectation, they behave like class B metals such as mercury. So here you have a graph showing the change in concentration in the cooked meat, whether fish muscle or seal liver. On the upper graph, it's the fish muscle, and we were not able to detect in that case the rare earth elements in fish muscle. I've explained before that it's hard to measure. But we could see that mercury was increasing because of loss of water, so that makes sense. And usually the class A metals, such alkaline earth and alkaline metals, were on the right side. So that made sense. But in the liver of the seal, we had rare earth elements, and we were expecting them to be on the right side of the graph, but they were not. They were on the left side, so with mercury. So it seems that they increased in concentration when we cooked them. So they have a high affinity for strong complexes in the food that prevented them from moving in the cooking juices. So the main take home from this presentation are summarized here. First of all, REE research in the North should be done with communities. That's my belief. REEs co-vary and should be considered as mixtures, and we should also include uranium and thorium as also a co-transported elements. Aluminum, iron, and DOC are good predictors of RE in fresh water. Lichens and zooplankton are good potential monitors of RE, one for the atmosphere, the other for the water. The free ion model helps predict bioaccumulation, at least in zooplanktons. We've done it also with fish in the south. It works also a bit. RE bioaccumulate in liver, but are quite low in muscles. And vertebrates accumulate REEs in inert fractions, but they accumulate a lot. And fish accumulate REEs in sensitive subcellular fractions, but they don't accumulate a lot. And the REEs are biodiluted along the food web in freshwater, marine, and terrestrial food web. And contrarily to what was expected, REEs tend to stay in food during cooking. And I would like to thank people who funded this research and people from the Kanji community, the Nunavik Research Center, the Malarijet Group, Nunavik Parks, and the Kativik Regional Government.